Hello and welcome to another episode of the Project Purple podcast. I'm Dean O'Reilly, founder and CEO of Project Purple. We have an exciting interview coming up with a very special guest after a few quick updates. Since 2010, we've contributed over $3 million and counting to pancreatic cancer research efforts. We've also allocated over $1 million worth of aid to more than 1,300 families and patients through our patient aid program. Your monthly or one-time gift can improve patients' lives through research and assistance programs. You can donate now at projectpurple.org. Pancreatic cancer does not rest, and neither will we. Your support is truly appreciated. If you're looking to get involved in one of our events, whether that means joining a run team, participating in a virtual run walk, group fitness, golf, or anything else you can think of, visit projectpurple.org/events to learn more. Without further ado, I'd like to welcome our very special guest to the Project Purple podcast, coming all the way from North Carolina. I had to think about that first. I was like, wait a minute, where is she sitting today? But Tobacco Road of all places. Super excited to have Dr. Kanu from Duke University School of Medicine, general surgery resident, and also super excited. I know we just put out the press release, so by the time this airs, the press release will be out there. But Dr. Kanu is also our Innovation Grant awardee for 2023. We just announced it here early. We made the decision in the first quarter of 2024. But super excited to have her here on the podcast. I've been fortunate to meet her a couple times on site visits down to Duke. And, you know, I say this, I say it often, but I say a lot of things. So I gotta gather my thoughts here as we record this. I don't have this scripted. So this is what happens sometimes, Dr. Kanu. I get you can tell I get excited for those that have watched this podcast in the past. I have the best job in the world. I've said this multiple times. I get to meet patients. I get to meet our participants who help us raise millions of dollars. And then I get to meet the researchers that are like you guys are on the front lines. I use a lot of analogies, and one of them is like, war. And I always feel like the researchers, the clinicians are on the front lines, like fighting pancreatic cancer day in, day out. And anytime, I had this vision of starting Project Purple and I said, like, when my dad passed away, like, I didn't want patients to go through, other families to go through what we went through. And how do we do that, is through, all the programs that we support patients, are helpful. But I knew day one when I had this idea, like, if we're going to change this, we got to do it with research. And so anytime that we fund researchers, and, as we've grown, we've been able to do more and more. And you're in this really cool time for us as we've grown tremendously to be able to, the first innovations grant for early detection. It just gets me so excited to be able to be in this spot as an organization. And then when you meet researchers like yourself, young, super talented, like super excited for the research you're doing, it just like invigorates us and inspires us so much here at Project Purple. So as I started, I said, like, I have the best job in the world. I get to meet all these great people, and particularly the researchers. Now, this podcast, like, when we had this idea to do this podcast, and I've always said, like, I've struggled, like, to articulate, just like I'm struggling right now to articulate how excited I am to have you on the podcast. But to have the podcast now to actually bring you on, and for you to be able to share all the great things that you're doing in this space, with our audience at large, and this thing will live forever, is just so cool. Like, I get so geeked out about it. So thank you so much for taking the time. I'm going to stop talking because I could probably ramble on for hours and hours, but super excited to have you on. We're going to talk about what you're doing in the pancreatic cancer space. We'll talk about the Innovations Grant, but let's get started first because hopefully you're going to share this with your friends and family so they know about your background and how you got to Duke. But for the greater audience that maybe this is the first time hearing, you speak, Dr. Kanu from Duke, hopefully they've read the press release so they know a little bit about the Innovations Grant. But here is your opportunity, as we said before we hit record, to kind of share your background, how you got to Duke, what you were doing before you got to Duke. And I will jump in from time to time and ask some questions. But with that, the microphone is yours.
Yeah, I can't say I have too exciting of a story. So prior to Duke, I was a medical student at Vanderbilt, and I actually remember my first year of medical school, one of the best lectures we ever had was just on cancer genetics, and I just absolutely loved it. The biology behind cancer is really fascinating. You can kind of think of it as a microevolution happening in real time. And through that, I got into cancer research as an undergrad, as a medical student. I was mostly studying immunotherapy, so one of the newest anti-cancer drugs out there, but unfortunately does come with adverse events. The patients, so immunotherapy works to increase your own immune response to cancer, but it also heightens your immune system to then create a lot of autoimmune events. So I was trying to figure out what are some predictors or risk factors that make patients at risk for that. Um, and then I was recruited to Duke, where Dr. Allen was actually one of my interviewers. And we really connected. When I matriculated here as a resident, he reached out to me, told me he was starting a new lab, the Pancreatic Cancer Research Lab, and if I would be interested in joining. And it just seemed like a really good fit. Again, very interested in cancer. At this point, I knew I wanted to do surgical oncology, so the surgical treatment of cancer. And he seemed like a wonderful, fantastic mentor. So I got involved in that way. And what makes Duke really unique, one, is its commitment to surgeon scientists or training surgeons who do want to do research. And two, so we have dedicated research time in my residency. So right now I'm taking time off from the clinical side of my training and just being 100% in the lab and developing my research skills there. And I also get to learn not just from the practicing clinicians like Dr. Allen, but also the basic scientists. So I have a cancer immunologist mentor. I have a cancer biologist and mouse modeling mentors. And I've really been able to learn the skills of how do you better study and model cancer. And one thing I've really been fascinated about, so I think pancreatic cancer is really a mystery in terms of how it grows. So I know I'm kind of preaching to the choir, but we all know it's one of the deadliest, devastating cancers. It's the third leading cause for cancer-related deaths, high mortality rate. And even in patients that present with the earliest stage disease, even after they go through the full length of treatment, so that's complete resection, negative margins, no evidence of disease remaining, also follow-up systemic chemotherapy, they're still at risk of recurrence. And the recurrence is the fatal event, not the primary tumor, but the eventual metastasis, the eventual recurrence. And because so many patients, even though we have completely removed their initial tumor, because they still keep recurring, the thought that I have, that many people are starting to have, is that, this disease is probably already metastasized by the time we already detected it. Even when it's just one small centimeter tumor, it probably is already disseminated somewhere. And so the question is, one, how do we understand how this disease operates? Because it seems to be different from our contemporary understanding of cancer. And how can we better detect that and catch that? So that's the space that we're operating in right now. And there are so many unanswered questions, so many unknowns in that space. And that's what I find really fascinating and really inspiring.
So if you look back at your schooling, so you did your undergrad was at I
Case
remember
Western.
Case Western.
Yes.
Then you go to Vanderbilt for your graduate medical, and then you find your way to Duke. So in all of that, was there ever, I know you mentioned immunotherapy, which like the at least for pancreatic cancer, we haven't seen the success that other diseases, I guess, have seen with immunotherapy. So there's this huge challenge, right? But prior, was there ever like the idea like, hey, I'm going to go into pancreas disease or GI cancers? Was there ever a time that, and I know hindsight's always 2020. I'm just always fascinated where this question's coming from, Dr. Kanu, is like, I always am curious, like, I know my reasons why I got into the space, but like, what is the motivation, the reasons for the clinicians, which I think is honorable and noble, and some of them are touched by it, but then others, like, it's just this really, really, really cool fascination with the disease. So was there a time in school that you ever like had an experience with PC or just something like in the lab that just like really kind of draw that interest?
I didn't have, so I had had experiences with GI cancers broadly as a medical student when I had elected to rotate on that service. I didn't have full exposure to pancreatic cancer until my first year of residency when I actually rotated on Dr. Allen's service and his colleagues as well. And that was when I actually got to care for those patients. And I think one, they really truly are special patients. They have a place in my heart. Just their spirit, their perspective is amazing. It also definitely puts things into perspective because many of these times, these patients go from otherwise living their normal lives, and then maybe they started noticing some yellowing of the eyes, some yellowing of the skin, maybe some abdominal pain, and then all of a sudden they get this diagnosis, and it's usually in such a very short period of time. And of course, they do their own research and they see all the stories of the poor prognoses, the high mortality, and yet they still have the resolve that, hey, I'm going to keep showing up. I'm going to go through all the treatments. I know surgery itself also has its risks, but I'm going to do all the things. And I think that spirit is certainly remarkable. So I really appreciated working with them in that space. I only did about 2 months of that my first year. And then my second year, we do a rotation at the VA, so the Veterans Affairs Hospital. And actually, it's a really unique position. As second years, we actually do run a little bit more of the cancer care. So part of our clinic is actually HPB focused. So that's hepatopancreatobiliary. So we'll see a lot of liver pancreas patients. And I didn't realize how much responsibility I'd have for that. So, for example, we have this multidisciplinary clinic where we discuss these patients with the GI doctors, medical oncologists, radiation oncologists, radiology, pathology, and surgical oncologist. And when we make a decision about a patient, we, the resident, then follow up with the patient, update them, and let them know what that decision was. And my first experience was actually having to deliver horrible news to a patient and working with him about next step, getting him back into the clinic and try to coordinate his care early on. And it does become personal when you start to have such an intimate relationships with patients. It definitely it definitely does impact you, but it also made it very meaningful. I did that twice and I really enjoyed that experience. And I think that was when I knew that, all right, this is the kind of physician I want to be. This is the kind of space that I want to be in. And I'm really grateful for that experience too. I don't think I had been as inspired about my career as a physician until I had those experiences. I hope that answers the question.
Yeah, no, that you nailed it. And I mean, I think it's inspiring to know, right? And I think I know we were talking about some topics before we hit record. I wish we had more clinicians like you, right? And I'm sure we do. But I am always curious. I mean, I think one of the things, the realities of this disease is that the outcomes are tough, right? And we are pushing, and I say we, I talk about the entire space from an advocacy standpoint. There's a lot of groups in this space, not as many as there should be. Let's put it that way. There always could be more, but I think everyone's really trying to push, research. The more research we can put into the disease, I think the better the outcomes would be, right? And I think that's one of the challenges that we've seen. I look at things maybe from a dollars and cents. That's why I decided to start Project Purple years ago, because I realized like there was so little funding, being done by the government and by private philanthropy. I mean, there was stuff, not to say that there wasn't, but I mean, when we look at numbers in comparison, to breast cancer, for example. And my mom's a 2-time breast cancer survivor, which is probably the reason why she's here today, is because of the efforts of the groups and because of the funding that has happened. But it's a stark comparison. It's like 1.2 billion compared to 200 million. Breast cancer is funding over $1 billion in research, and pancreatic is still in the low 100 millions. Which, again, the more research the more money that's available, the more research will happen, and these advances can happen. It's just awesome to hear, and I'm glad you're on this side and not in another cancer. And we're excited to be able to fund your research. So let's talk about that. So, as I mentioned in the opening, this was you're the first, and we hope to bring back the Innovations Grant again in 2024 and in late 24. But you are the first recipient of this Innovations Grant. So let's talk a little bit about this grant and what you're doing. Kind of and we can stay like high level, for the audience as well. Just talk about the aims and maybe where the idea and what maybe the probable outcomes would be from this.
Yeah. So as I talked about briefly, one of the issues with pancreatic cancer, it has very minimum symptoms. So as I mentioned, a lot of patients get diagnosed either incidentally, they came in for something else, they got a CT scan and it just happened to show it, or they were doing just fine and then all of a sudden they had a slight abdominal pain and then they got the diagnosis. And a lot of times, at least half of patients are already diagnosed with stage 4 disease. Or at the time of diagnosis, it's already stage 4. So at this point, we're really interested in the early precursor lesions. So you talked about breast cancer. One of the achievements with breast cancer is the ability to have these very fine-tuned surveillance mechanisms before cancer actually develops. Same with colon cancer. They've been able to develop this. With pancreas cancer, we don't really have that. The only lesion that we're actually able to detect before it's cancer is something called IPMN. So it's a cystic tumor. So cystic tumor, not yet cancer, but it's just a mass. And we know that there is a risk for cancer development with these. We're able to surveil them until they start having high-risk features, at which point we would recommend that they undergo surgical resection. The issue with that is so surgical resection is the Whipple procedure, which is the same surgery that we would give to a cancer patient just to remove that part of the pancreas, or also distal pancreatectomy, depending on where in the pancreas it is. Sorry, that's more detail than needed.
No.
Pancreas surgery in itself also has high risks associated with it, high morbidity. So we often have to weigh, all right, what is the actual risk of cancer that we're saving this patient from versus the actual risk of this procedure that we're subjecting the patient to. So our big focus, among many, but one of our major focus as a lab is trying to figure out what are ways that we can actually stratify these cystic tumors. Can we detect any biomarkers or any kind of features that say, yes, this tumor is a heightened risk for cancer, so they should proceed to surgery, versus this tumor is lower risk, so we're fine with just surveilling them. And for that reason, we have two aims. So the first one is actually studying human tissue. So of all the patients that we've currently resected, which of these tumors, we looked at them in the path. We've divided them up into low grade versus high grade versus tumors that actually were found to have cancer in them. We're sequencing them to see, are there any genetic features that differentiate them? And can we then use those genetic features prospectively to kind of risk stratify patients moving forward? And then the second aim of that is actually developing a mouse model of IPMN. There are mouse models of that, but they're not as genetically similar to patients as we'd like. Many of them don't use the same mutations that patients have. So we're trying to create a mouse model that actually uses the 3 main genetic mutations we see in patients. And then we want to actually follow out these tumors to say, all right, as they're separating, what are the low-grade lesions? What are they composed of genetically versus the high-grade lesions versus the ones that do develop mild to invasive cancer. That allows us to actually track the development from normal pancreas to low-grade lesion, high-grade to invasive. And we're not able to do that kind of longitudinal surveillance with patients at the moment because we're just limited to when we were able to get the tumor that one time point.
So I just want to back up here for the audience. So IPMNs are fairly common, correct? In just the normal population.
I'm sorry. I guess it depends on your definition of common. I wouldn't say like the average person has one.
Yeah. I guess like then the question. So I mean, I guess I relate this, and I know we've talked about this in the past in person, but like so. When we think about like early detection, we think about like risk, like IPMNs within certain communities, or I guess in certain population, like aren't as prevalent, but they're there. But the bigger question is like, why do some become cancerous? Just because you have an IPMN doesn't mean that you have cancer. Just like so, where I'm going with this is like we talk a lot, and we've funded a lot about genetic risk, right? Like we know with certain genes, like there's a risk, right? Like there's a risk, like just like with breast cancer, right? Like if you have BRCA, and so with pancreatic cancer, if you have the BRCA gene, for example, which is probably the most common, like, yeah, I think the general population risk for pancreatic cancer for a male is like 0.4, I think, or like, it's like 0.3. I forget what, and don't quote me on the numbers, but I know it's like relatively low general population risk. With the BRCA gene, now it goes up to, I think, to like 7%, which again, relative to population is low, but it does increase your risk. And, now like, if you have a BRCA gene or one of these genes that we know, like you should be in some sort of surveillance, at a certain age, especially if you have a family history. With IPMNs now, we're realizing like, hey, like certain pockets of this population have these IPMNs. Doesn't mean that you're at risk of getting cancer, but again, you're at a higher risk and these things should be monitored. But they are relatively common within the pancreatic cancer space, I guess is where I want to go with this, is like, we're seeing a lot of pancreatic cancers start as IPMNs and then evolve into cancer. Why that happens, we just don't know.
Yeah. So I think the question you're raising is why do some progress to cancer, right? And why do some stay where they are? And that is the fantastic question that all across the board of pancreatic cancer research we're trying to grapple with. If I can do a little bit of a segue. So there's IPMN. There's also something called Pannin that's conventionally thought to be the main precursor of pancreatic cancer. Although recent research is finding that Pannin is prevalent even more so than IPMN, that you and I probably have tons of Pannin in our pancreas. And so why is it that you and I are still doing just fine, knock on wood, but somebody next door to us got cancer? And so then that becomes that whole nature versus nurture, where the question is there something about the environment of the pancreas? Is there something about inflammatory insults or something else that increases the risk besides just the presence of these precursor lesions? So a lot of people have explored, well, what is the role of chronic pancreatitis? What is the role of chronic inflammation? What is the role of diabetes?
Correct.
Answer those questions.
I think like bigger picture, like, so I mentioned genetics, we're talking about IPMNs, you mentioned pancreatitis, diabetes, right? Like, I think this is just a statement. I think with this space, like there have been, and I do think so I've been here 14 years, there have been some advances, right? It's just not clearly where we want to be. But I also feel like, and I remember talking to one of the researchers, I remember in like 2018, I was at a AACR pancreas meeting. So it was just for pancreatic cancer up in Boston. And it was Anirban Mietra. He put up this pie chart and he's like, in 2006, we had, these 6 categories. And here we are, fast forward in 2018, the pie that had 6 slices is now like 22. right? And his point was it just, as we learn more, it gets complex.
Yeah.
But the key though is here we have to understand this, right? Like we have to understand IPMNs just like we have to understand genetic risk and diabetes cohort. So I think all of those are important. But as we understand the disease, I guess it's a testament to, again, researchers like yourself doing the work. Like we've got to do the work. That's something that I always tell, other families is like, we've got to get funding to these researchers to actually do the work on these studies that, again, not to compare, I hate to compare, but we have to, like breast cancer, they've done the work, they've done the studies, they've had drug development. We just haven't seen that here in the space. But I do feel like there's so much that's happened in the last 5 years. I think maybe everyone, maybe, coming out of COVID, there was so much energy and positivity that, hey, now we're back to normal and, events are happening, people are gathering and, oh, research is happening again, like there's funding out there where I think during that COVID year, everyone was kind of like in this standstill. And, I know a lot of the federal funding went to COVID research, but, I think that that's maybe some of the excitement, but there are a lot of things happening. And so we're super excited again to be able to have this opportunity to fund some great research here that you're doing in this IPMN space. Talk a little bit about, I know you mentioned, having the background in immunotherapy and, we haven't seen, and this is maybe just a personal opinion, not to, I guess I am putting you on the spot. I was going to say not to put you on the spot, but I am putting you on the spot. What are your thoughts about immunotherapy? Because that comes up often, and that we'll take it, from there. Like, why haven't we seen kind of some of the advances we've seen in other cancers that we have not seen in pancreatic cancer?
Yeah, yeah. So immunotherapy, sorry, I'm gonna back up a little. So how immunotherapy works is it basically revs up your immune system to clear cancer. So the average person is forming cancer cells all the time. That in itself isn't new. It's just that our body is really good at getting rid of them. So cancer develops when it's able to evade those mechanisms, one of them being immune clearance. So immunotherapy works by basically inhibiting the turn off switch of the immune system. So it's just constantly on. But the sorry, I'm trying to take this to not get too lost in the weeds. How your immune system recognizes a cancer cell or a virus or a bacteria or any defective cell is through neoantigens. So these are proteins that are
Right.
expressed on the cell surface that tells the immune cells that, hey, this is a foreign protein, this is or abnormal protein, an abnormal cell that needs to be extinguished. So for cancers that have a lot of neoantigens, like melanoma, like lung, this works very well. Pancreas cancer tends to not have a lot. It's something called a low mutation burden, so it doesn't have
Mhm.
a lot of expression of these abnormal proteins. And the tumors themselves don't have a lot of cells to even express those proteins. And also, the tumors tend to be surrounded by these dense fibrous stroma. So, just like lots of, think of it kind of like scar tissue. So, it really inhibits immune cells from penetrating into the cancer itself to actually combat these cells. So, that's where we are currently and why we're limited in that realm. Um, there have been some ideas that I've tossed around with some folks to look at. All right, so if you compare patients that have actually high cellular tumors versus low cellular tumors, does that lead to differences in outcomes? Just a thought that I had. If like maybe the one tumors that have more cells are therefore able to have more neoantigens, therefore able to increase recognition by the immune system. I'm sure other people have studied to that more.
I love it though. So I think you did an amazing job of explaining that, and I think that that I I guess for the audience listening at home, if they're the people that are in this, I think that was perfect. I know the media, we get calls all the time, right? Like, there's this new drug, it worked for, XYZ. Or I think in this space, we've seen very little success within immunotherapy. And I remember like 60 Minutes did a segment, I think it was probably like it was right before COVID, and it talked about immunotherapy. And there was one patient who had pancreatic cancer. So then our phones like rang off the hook, right? And so I think It's an interesting subject. I know again, as I go back to what I said previously, I think the more research that groups can fund, hopefully the more success that the research community as a whole can have. With hopefully using immunotherapy. I know it's just been a real big challenge, and
Yeah.
I think there's been certain parties like the media, as I said, that have maybe promoted stories that probably aren't 100% accurate in terms of the effectiveness of immunotherapy within our space. So as an advocacy group, it becomes really frustrating because when those stories do go out, I think there was another one that was like a publication. It was a group in Europe. And that one, our phones were ringing off the hook, people trying to get into, particular clinical trial, or, is this actually true? Does this work? And that one was just in mice, of all things. Right. But the media took it and ran with it a certain way. And so, yeah, it's frustrating, as an advocacy group. But again, this is the reason why we're doing what we're doing, and the reason that, whether it's us or other groups, need to continue to fund, talented researchers like yourself, and continue to keep pushing for more funding from all sources, not only philanthropy, but government and, hopefully pharma. Couple of questions here for you left, and then we're at the end. We're going to share, hopefully, if there's a funder out there, or if there's people that want to learn more about all the great things that you're doing, where they can learn more online. Where do you see yourself? I'll back up. I know you still have residency at Duke, so this is the next couple of questions I always say are loaded. So there's no right or wrong. But let's say we go down. We grab a crystal ball. Where do you see yourself in 5 years, Dr. Kanu?
Oh, 5 years. So actually, the nice thing about medicine, we have this whole timeline. So just disclaimer, I am fully committed to surgical oncology, well, we call it HPB, so hepatopancreatobiliary. That
Yeah.
is where pancreatic cancer is housed within surgical oncology. Um, Overall goal, so 5 years from now, I will hopefully have finished general surgery residency and would probably be halfway through my surgical oncology fellowship. The broad goal for that would hopefully have a career like Dr. Allen, in which I would be a surgical oncologist specializing in pancreatic cancer, also running a laboratory of my own. So being able to be in both spaces of that of academic research, basic science research, but also in the clinical care of patients. I think that would be the most fulfilling to me. I love cancer research. I find it absolutely fascinating, intellectually stimulating. There's so many questions and ideas. I was kind of restraining myself from going off on all of these different ideas I wanted to share with you, which we can talk about if you want.
Yeah.
But also, I know I can't 100% be in the lab because I need the patient care aspect. That is what's really the most meaningful and satisfying for me. So a career where I can do both of those things would just be the perfect fit for me.
I love it. And, I just want to say something. So, like, there's no right or wrong. I don't want this to come out like sound like jerky, but like, I think that I've met, in 14 years, some really, really amazing surgeons. Then they love to just operate, right? And then there's the flip side of that, which there's no right or wrong to it. And I think the world needs both. Is you have people like yourself, right? Like, you love that clinical aspect of being able to do surgery, but then you have labs, right? And you do a lot of research. And I always say, like, research is where the changes are going to happen. And some of it will happen in clinic, right? Like, I think like people doing, volume, is important, because practice makes perfect. But then, some clinicians just don't, they don't want to do research. Like, that's not their jam. And, they don't want to, get involved in that space, which is okay. There's no wrong to that. But I do feel like the research aspect is really critical, especially where we are in this space, to really move the needle. And the other thing that I have not mentioned, um, you're young. So it is so exciting. When I was at the last, I think 2 years ago, I was at the last AARC meeting for pancreas cancer. There were so many young people in the audience. I mean, there's some dinosaurs in this space. Like, and I don't know, like that's great. Like experience is great, but like the future is the youth, right? And so when you see like so many young people that have so many great ideas and that are committed to this space, it just again is inspiring and gets you really excited for the pipeline and really for those patients that will be impacted by this disease in the future to have so many young people heavily invested in this space. So thank you. So on the question, I said 5 years, and I know you've got a lot of schooling left, your residency, surgical oncology, and for those that have been keeping the math at home, I mean, so like when you started, let me ask you this question. So from your undergrad to when you will be done with surgical residency, like how many years will that be? Like from start to finish?
Oh man. So this is the depressing part. So 4 years of undergrad, then 4 years of med school, and then 8 years of residency. So it's 5 years of clinic. And then I'm going to do 3 years of research time. I extended that time because I really want to invest in it. And then I'll do 2 years of fellowship. So that'll be
18 years.
yeah.
Wow. So
Yeah.
I mean, so we're laughing, but this just goes to show the expertise. I mean, in 18 years, of schooling, and I mean, and like we're going to say, well, granted, 4 years was undergrad, but it's still 4 years is college, undergrad. So like, just that's you think about people talk about like this 10,000 rep rule, right? Like you have to do 10,000 reps to become like Michael Jordan, the GOAT, right? Where you're into back. Oh, I shouldn't bring up Jordan. Maybe Coach K. That's a better analogy for someone at Duke, like Coach K, like, he's one of the winningest men's basketball coaches of all time, right? And, you talk about reps in order to get all those reps in. So that experience and the things that you do over that time, again, super excited to have you in the space. We will do whatever we can at Project Purple to keep you in the space. I'll just put it out there, but super excited. So my question here, which relates to like, where would you be in 5 years, but now also your experience that you've had, and let's play make believe here. If you had the ability to start your own lab,
Mhm.
and let's say I'm going to give you a blank check of like $15 million. So money's not really an issue because I think in today's economics, I think it takes like anywhere between 15 and 20 to like open a lab, right? To get it set up. So let's take the money equation out of it. But given your experience up until this point in time, like where do you focus for pancreatic cancer? Like if we want to try to increase survivability, where would that be given what you've done and what you've experienced?
Yeah, so I am really fascinated by lineage tracing and cancer development. And that's partly because of the cancer biologist that I work with. He's kind of been in my ear about that. So, brief tangent, my mouse model of IPMN, how it works is each of the mutations that we've engineered has a fluorescent barcode. So it, in addition to expressing the mutation, it also expresses a fluorescent protein with their own colors. So then we can follow out these mutations as they develop and we can see, oh, this tumor is mostly yellow, this tumor is mostly green, or not green, sorry, blue, this tumor is mostly pink. Or maybe it's a mix of those, or maybe it's just yellow and blue or all pink? And are there differences in behavior that way? So, what I would love to do, we know that KRAS is the dominant mutation, driver mutation in pancreatic cancer, but there are different kinds of KRAS. So there's KRAS G12D, KRAS G12B, KRAS G12R, and each
Yeah.
probably each have their own, I guess, oncogenicity, have their strength of tumorigenesis. So I would love to have a model where we have these different KRASs with their own fluorescent protein and then grow them out to see, all right, how do they affect tumor behavior? Is KRAS G12D just like proliferating everywhere and all those mice die? Or is KRAS G12R, maybe it's silent, but it metastasizes earlier. So before we even see a tumor, there are already metastases in the lungs and the liver. We could see those too when we euthanize the mice. We can look to see, oh, okay, we saw a yellow cell a yellow cell here in the liver, but we actually haven't seen anything yet in the pancreas. And I think that would really get to the question of how is that early stage behavior in the pancreas? Does it actually kind of start as a tumor and then kind of grows, and then metastasizes? Or does it metastasize early on before we can even detect it? And that's the question that I really would love to answer.
You bring up something in just in the last like 2 months, we've had so many discussions on the podcast and off about like these mutations, right? These KRAS mutations, and you mentioned many of them. And I feel like this is something like in the last 5 years, because like 5 years ago, I didn't have patients emailing me saying, hey, like I have this KRAS G12C or G12R mutation. Do you feel like, again, this is a loaded question, but in your experience, do you feel like that's like kind of the direction where, and not just with the KRAS, but also like we've seen BRCA, right? Like with the genetics, do you think like we're moving in this space for PC where we're getting really refined in terms of like the mutations and where these things start and how we're classifying certain patients? Like, do you think that will be commonplace like as we move forward in like the next 3 to 4 years?
Yeah, so we call that precision medicine, and that's
Correct.
something that's really taken the whole cancer field by storm. And as a plug, that was started by Vanderbilt with the BioVU, so that's my legacy. I do think that we are moving in that space. Clinically, the benefits of that, I think we're cautiously optimistic. I know that's a cliche term. I think the long-term goal of that is that you can have specifically targeted drugs for that patient's mutation, right? So we're trialing out KRAS G12D inhibitors, KRAS G12C inhibitors, pan-KRAS inhibitors, so inhibitors of all the KRAS mutations. The problem that we're having right now is the early data seems to indicate that patients do get eventual resistance to that. What that means, I actually have to parse that out if it's that they still have that same KRAS, but it's no longer responding to it, or if they develop a new KRAS mutation. I think that's probably what makes more sense. They've just developed a new KRAS mutation that's now not responding to that drug. And that's also kind of why I want to do that lineage tracing experiment so we can actually figure out how those different mutations play out. So to your question, I do think we're moving in that direction. I think there's a lot of hope and promise with that. I think it still needs to be realized, the full potential and the full benefit of that for pancreatic cancer.
I love it. I love it. And I think to your point, though, and you just said it, like we still have to understand more the biology of this
Yeah.
disease, which again reiterates the need for further funding. So for those listening at home and watching, like this is why research is needed. And this is why, Project Purple, why we've been on this mission to continue to fund extremely talented researchers like yourself down at Duke, and we're super excited for that. So thank you for sharing all the great things that you're doing, all the things that you will be doing in the future. Hopefully, we'll continue to support that effort. My last question here, and then we're going to share kind of where the lab, where people can learn more and take a look. But this is always the last question. I think it's always kind of interesting to get everyone's perspective. There's no right or wrong here. Given your experience, what you've gone through, how would you define the term pancreatic cancer? How do you define it? Like, what does that word mean to you?
Mm. Yeah, so uh, the obvious answer is, well, you have a malignancy of the pancreas. I do think that. I think it is a cancer cell that has developed abnormal mutations to allow it to grow, but I'm also becoming increasingly a believer that it's also a deficiency of the surrounding pancreas in its ability to squelch that abnormal cell. So I think it's both. It's both an abnormal cell and also a defect in the remaining pancreas.
I love it. I love that. I love that definition. And I love that you kind of bring in the work that you're doing and where you've seen it from your aspect of working with this and being in the trenches. As I mentioned before, I consider our researchers and clinicians really on that front line. So Dr. Kanu, thank you for all the work you're doing. Thank you for the insight. Thank you for the opportunity to have you here on the podcast. We are super excited to be funding your work and hopefully to continue to fund the great things you're doing down there at Duke. If someone listening or watching wants to learn more about the great work that you're doing on there, where's the best place? I know we talked a little bit, like there's a lab page or maybe the web.
Yes, so there is a newly released or revamped lab website. It's the dukepcrl.com. So Duke Pancreatic Cancer Research Laboratory.com, dukepcrl.com.
Awesome. Thank you for, again, all that you're doing. It's truly an honor to support your great work. And thank you for being a guest here on the Project Purple podcast.
Pleasure. It was a lot of fun.
Awesome. Thank you for listening to another episode of the Project Purple podcast. If you like this episode, feel free to share this episode. Make sure you follow the Project Purple podcast wherever you listen to podcasts. That's a wrap of another episode. Please be safe and thanks for listening.